Hello and welcome, everyone. My name is Joanna Niska-Blakie, and I will present the guidelines for reagent choice in the development of diagnostic tests. I will start with a brief overview of BLIRT. We're a Polish biotech company manufacturing enzymes and proteins used in molecular biology. Our motto, enzymes as you need, very well reflects our business philosophy, how transparent and how flexible we are as a company, as well as our customer-oriented approach. We started as a technical university spin-off over 25 years ago. Then we became founded in 1994 and then visible on a recognized stock exchange since 2011. Among with the large-scale production, we are dynamically growing as a team, and currently BLIRT employs over 80 people. And what is important for those of you that develop diagnostic tests, we are both certified with ISO 9001 and ISO 13485. I think that we all observed the significant increase with regard to point-of-care facilities. And there are multiple type of tests in POC. So first of all, there are molecular tests, which are known for the high sensitivity. They allow to detect of genomic variants as well as the right diagnosis. And what is important, they help to monitor the patient's response to therapy. And beside molecular tests, we do have antigen and antibody tests. The antigen tests detect the antigens expressed by the virus. The antibody test, in opposites, they detect the antibodies in the blood. And the common features of these two latter tests is that they're cost-effective and they're very rapid. There is a rule for those of you, especially for startups that develop diagnostic tests, to narrow down the target market, meaning to focus on a certain type of molecular, certain type of test. And I would say that the same rule applies to enzymes manufacturers, as for instance, BLIRT produces the enzymes that are used in molecular diagnostics. And in molecular diagnostics, we can distinguish numerous methods. The gold standard is obviously the RT-PCR. And BLIRT manufactures enzymes that cover every single step, starting from the sample preparation, when you need proteinase K, when you're using the RNA isolation kits, with a very important step, which cannot be overstated, which is the RNA isolation protection with the RNase inhibitor. And then going through the genomic DNA removal with the specific double-stranded DNases, reverse transcription, and finishing on the DNA amplification. More and more often, there are other methods that do not need sophisticated instrumentation. And such example is the RT lamp. And when you look at the enzymes, they are quite similar to those that I was talking about, where you use during the RT-PCR. But obviously, there's a different polymerase being used, which is the isothermal BST DNA. RT-qPCR and RT-LAMP are methods which are frequently used during the detection of viral infections, but there are other methods, especially used in cancer diagnostics. And I would like to give an example of in situ sequencing, which basically is sequencing of the mRNA in a section of fixed tissue or in a cell sample. And beside the standard enzymes and proteins being used, there's the need to use DNA ligases such as TTH or T4DNA, P29 polymerase, as well as RNase H. And I would like to give to those of you that think, doesn't matter if you're a startup or an established company, think of development of diagnostic tests and give you the few very important guidelines, which are based on the most important features of enzymes that you always should to look at. And we group them into 4 main categories, which is the high quality, stability, reagent compatibility, and availability. And I will go one by giving the adequate examples. So the first big group is the high quality, which defines sensitivity and specificity of the test. And in this group, we distinguish the high competitiveness of the reagent as well as batch-to-batch consistency. So when it comes to high competitiveness, when you're looking for a supplier with a certain reagent, you would like this reagent to be comparable with these ones from the top market leaders. And it's always very important to define what is the most important feature that you're looking in this reagent. So let's imagine that you're looking for a proteinase K, and the minimal activity that you need for your assay of proteinase K is 30 units per milligram. And the activity, what you see on the graph, is indicated by the blue line. So when you find a supplier, it's always very important to think of the alternative one, because in terms of certain circumstances that you have no influence, such as the shortage of the supply chain, you need your business to keep running smoothly. So it's always good to know in advance what are the reagents that you can use interchangeably, not to affect the specificity and the sensitivity of your assay. Another important feature that I would like to mention today is the batch-to-batch consistency, because you need to be assured that the product that you get, its features that are declared in COA won't be changed from batch to batch. So because the low variability equals high reproducibility, and you need your assays to be reproducible. Another important group that is definitely worth mentioning is stability. Stability in terms of shipment, in terms of different temperature during reactions in your assay, and as well as stability of enhanced stability of different products, of different formulations, such as a lyophilized product or lyo-ready products. So stability during shipment is very important. And nowadays, you can find more and more products that have enhanced formulation that allow the shipment at temperatures different as -20, for instance. And this is an example of proteinase K in liquid, where you can see in comparison its activity, which here is indicated the minimal activity as 800 units per ml. You see that there's no difference up to 18 months when proteinase K was stored at 37 compared to 4 degrees. So it has numerous benefits. Mostly it's cost effective. Also, it doesn't need you to store it in an expensive freezer in your laboratories. And what I also meant by stability is that stability of the reagent during different temperatures of the reaction of your assay. And here, a very good example that I would like to share with you is the RNase inhibitor, the RiboProtect. So on the first graph, you see that the RNase inhibitor was incubated with RNA and RNase A at 37 degrees up to 6 weeks. And what you see that the activity was not changed until 4 weeks. Only it decreased by 20% by week 6. But when you look at the higher temperature, which is 50 degrees, you see only 50% drop by 48 hours compared to 24-hour, where the activity was 100%. So it means that the reactions, the processes can be carried at different higher temperatures, and it gives you a guarantee that there is no need for enzyme readdition, which again is cost effective. And another very important feature, and I would say it's a real game changer in molecular diagnostics, are products with a different type of forms, which is the lyophilized form and the lyo-ready form. Lyophilization is a process of freezing a product in a solution, then placing it under the vacuum and removing the ice through sublimation. And in this way, we end up having a powder, which is very stable, which doesn't take much space. So it's relatively cost effective to be shipped as well to be stored. And the most important feature of such products is that they remain sensitive and highly active after being reconstituted. And the other products which become more and more popular in diagnostics are the so-called lyo-ready reagents, which are reagents in solution, but they do not contain glycerol, which is a very well-known factor, stabilizing factor. And such products are compatible with lyophilization, are compatible with numerous additives, what means that after lyophilizing and reconstituting the entire uh, set of products, the mixture of products, they remain sensitive. And such an example is the riboprotect, the RNase inhibitor. The third very important group of features that I wanted to talk to you today about is the reagent compatibility and test optimization for clinical use. So, as a company, we do not perform test optimization, but what we do care for our clients that purchase products from us is that altogether the products, after being, for instance, lyophilized or reconstituted, they remain sensitive and specific. They remain the same efficiency as they had before being colophilized. And this is an example coming from our client who was developing the RT-LAMP method, and for which he used the RiboProtect lyo-ready RNase inhibitor, the transcript me reverse transcriptase in a lyophilized version, as well as the BSD polymerase. And what important fact is that after lyophilization or reconstitution, he noticed that the reaction to assay was, uh, when it comes to the efficiency, very satisfactory, and that they all worked well together. So there was no cross-inhibitory effect noticed. When you're developing tests, you can also manufacture certain enzymes by yourself, but you should keep in mind that there are certain products, such kits, during the isolation process or during amplification, that you can always get as a ready-to-use product from other suppliers, being aware, being sure of the compatibility. So this is a big facilitation that saves your time and saves your money, and trusting others' expertise is very beneficial also for you. And the last group of features that I wanted to present to you today is availability. Availability of the product and the production, the size of the production, the worldwide supply, and the product adjustment. So when you're looking for a supplier, it's always worth searching for one that has established and stable supply chain, as well as the large production scale. It means that you can count on a bigger amount of products coming from the same batch, which again, it's a very positive factor um, for, let's say, the consistency, for the low variability in your assay. So it's a very important feature always to pay attention to. And when you're developing an assay, there are different types of steps that you should take. So first of all, always consider different types, the variety of forms of the reagents, and try what suits best your business and your assay. So products in different forms, such as cake, liquid, powder, or the lyo-ready version. Once you decide which reagent suits you best, suits your assay best, you should always talk to your supplier about the possible product adjustments, such as like fermentation adjustments, some characteristic change that you think might uh, suit better your test. But it also could be that as a manufacturer, you already developed a fantastic product, but what you don't have is the capacity, the manufacturing capacity. So this is the point where you should always look for a partner with an OEM option. And at the same time, a partner who can offer additional services, such as labeling or aliquoting. So these factors are all very important in your business development. So I'm approaching to the end, and I would like to leave you with a kind of a home take message, how to strengthen your communication with the supplier. So it all starts with an idea in your head. When you think of certain tests and reagents that you need to develop it. So first of all, you're looking for suppliers, you contact several. When you contact a supplier, you should always ask for a sample, ideally for a few samples, so you can check the batch-to-batch consistency by your own. It's always worth to sign the NDA in the meantime, so you can talk to your supplier more fully, so you can discuss more details of your assay. It's always beneficial for both sides. So once you are happy with the product gave you good results, it's the time that the product goes through the QC development and evaluation. Then there's the final product approval. And again, to be on a safe side of the business, it's always worth to think of signing the supply and the quality agreement to be sure that there won't be any changes in configuration, for instance, of the product formulation during the time of how where the agreement lasts. So this is all very important factors because any change can affect your assay at the end. And from that point on, you can be very happy running your business, having the stable supply chain.